Okay.
So as I said, we've developed games, workshops, and events for all ages from 9. We have done a lot of things with small kids from 9. And these activities were developed since 2000. Most of the time, that's to follow the discussion. It's either you only talk about a biological context and you try to introduce biological databases and bioinformatic tools within this biological context, or you just try to teach about the biological database and the bioinformatic tools. For me, as you say, the curriculum is very important. So for me, it's better to try to have several different biological contexts, several different stories. And in each of these stories, you always introduce quite the same biological databases and bioinformatic tools. For me, it's the best way to do the things. So the teacher will become familiar with the tool, even if the context is completely different. And for me, that's the way to introduce bioinformatics to the high school teacher. And then they could take the material and try to figure out a new story, a new biological context for their own curriculum. That's my opinion. I don't know if it's the same for everyone, but that's the way I'm working. So each time I am choosing a biological context, which is most of the time related to a peer review article, and we try to give access to the real data to be able to redo the experiment from the article. And all the time, we try to do computer activities, but first we try to do pen and pencil approaches. So they do something manually, and then they have to do the same thing by using the computer. So each year we have about 3,000 participants, and it means that more than 2,000 students from 12 to 18 years old. And the teacher are always here when we give activity with the class. So we have teacher coming each time. But we also do specific session with the high school teacher, private and public schools. And this is once or 2 times a year. And we reach about 30 to 50 people. It's not a lot, but okay. We have not so much people to reach in Switzerland. Everything, all the material we have is online on this website, which is mainly in French. Okay, we get the problem of the language, but some of the material has been translated in English. And it's very funny when I see the guide on the detective food, because a long time ago, we have developed the pizza metagenomic, which is a biological context to introduce metagenomics. So the people have to identify the different species present in a pizza based on DNA. So they do BLAST and they find the new species. And we have adapted this workshop to another type of biological data. It's the hair. So they had to, the kids had to know if they were in the campaign, in the mountain, or in town, if they were or not epidemic because of the virus present in the samples. So this type of workshop has been published in the EMBnet journal in 2016. And I was very happy to see that some of the ID were taken in the food detective. It means that we should bring together all our ID, because yeah, there are very nice ID everywhere on this planet. And as Terry said, in 2014, we gave a workshop together with Patricia Palagi for high school teacher. And the material was prepared for that workshop and was translated in English for that workshop. And it was dealing about how you can better understand genetic disease with the help of bioinformatics. So as Terry said, she had to deal with the crude material, which was prepared for online and face-to-face presentation. And the results is this guide, which I'm very happy to see that he has a nice, the statistics are nice, and the click and how many people have been looking at it. And more recently, together with my colleague, we developed a workshop on drug design. The idea was to show the people how you can design new drugs with the help of a computer. So we were focusing on the treatment of cancer and pain, and we give access, starting from a very nice and easy website, we give access to professional bioinformatics tools which are used by the professional. So Terry has been working a lot on this workshop to get together with us a nice practical guide. So this workshop was published in the Journal of Chemical Education, also published in another journal, Chemical Education, and based on this article and on the presentation I gave to Terry, she was able to write the practical guide. So we are, as she said, it's coming soon. We are nearly finished with the guide. So the goal of this guide was to show the people how you can design small molecules by drawing your own drug candidate, how you can compare the protein that target, the strength between the protein and the drug by using docking bioinformatics tools, how you can calculate and predict the properties of the drug candidate and infer if they are toxic or not, if they reach the brain or not. And if you can predict which protein will be targeted by the drug candidate. This is the website here. It's really an iterative process. That's the way we want to show this workshop. So you have to draw your drug to see how it's docked in the protein target. You have to estimate the binding strength and compare with existing drug. Then you have to look at the properties of the drug. Does it target the brain or not? Is it toxic or not? Using a predictor, which has been developed by my colleagues. And also you can predict which protein is going to be targeted by the drug. So we have, as I said before, we have 3 workshops, 1 on the pain, 1 and 2 on cancer. And for the guide, we decided to talk about the pain and more in detail. We decide to modify ibuprofen to see if we could improve the drug quality and the properties of ibuprofen. Always by using 3 professional tools. The first one was the docking system, which allowed to calculate how the drug go into the protein and what are the strengths of the binding. The toxicity, if the drug is reaching the brain or not, and if it is specific or not to the protein we want to target. So I'm quite happy to say that the guide is very good, and thanks to Terry for her energy and input. Just to let you know, more recently I've developed a workshop on the coronavirus, of course. And the idea is to look at the data which are available for the coronavirus, the 3D structure of the spike protein, the genomic data from different humans on this planet, or the genome of the virus from different mammals. And one of the questions of this workshop is how to find the origin of the virus. So we provide some sequence, and the people can align the sequence, and they could have a look at the tree and make hypotheses on which mammals which virus from which mammals was the ancestor of SARS-CoV-2. I also use the article and to know how you can discuss about the fake news coming from this COVID disease, such as that the virus was not natural and because it contains some sequence from HIV. So we have developed some activities to show the youngest that you can take a piece of DNA, do a blast, and tell the answer you want. If you want to say that the piece of DNA comes from HIV, you can do this. Even if you change the letter in the sequence, you will find the same results. So it's a way of illustrating how people are manipulating the data. And I find this example very good because it's really one of the examples in the social media where they say that this virus was not natural. So maybe, I don't know if Terry has the energy or not, it could be another guide. And if other people have been also doing some activities on the COVID, maybe we could bring together all the idea we have to construct a new, a new practical guide. And for those of you know the website we developed a few years ago, Chromosome Walk. Now we are doing a new version, which will be a smartphone responsive, and we have added a new section on precision medicine, explaining why we need now NGS in medicine. So by taking the example of the mutation found in BRAF and how you can decide the treatment of melanoma if you have or not the mutation, we have developed also a workshop, and it's always the same. The people have to analyze the DNA sequence, they have to translate using some tools, they have to do blast, they have to look at the position of the sequence in the human genome. They can do multiple alignment to look at the conservation in other species. And also for this workshop, we can go then to the drug design, because it's the example we gave for designing a new drug in cancer by using the mutation found in BRAF. We have developed also some material such as 3D printed proteins, when the people can play and try to put the drugs into the protein with or without the mutation. We have developed puzzle where the youngest can construct a molecule and try to figure out which molecules can go into the good proteins. And also we have printed out the sequence of chromosome 21, and this is a very, very nice tool to explain the big data and how difficult it is to manage all these DNA sequence. So the workshop about the precision medicine could also be an idea for a next guide, if Terry is agree, and if other people have also other example in the same field, this could be very nice to bring all the material together. So for me, these practical guides are very useful, and thanks again, Terry. They allow to unify and standardize all the material which is already available among GOBLET members and even more members. We can reach World Wide Web high school teacher. I'm just wondering if it could be possible to better communicate about these guides and to put it on other websites such as Scientix, or I don't know, other website dealing with a high school teacher. And as I said, and we said before, to translate this guide in other languages, maybe this could be sponsored by GOBLET, I don't know. But language is a really, really big major challenge. I will say even more than the curriculum. The language is really difficult. If you have only material in English, it will not work in Switzerland for the high school teacher, for sure. So thank you very much for your attention. And if you have any question, I'm here and very happy to answer. Thanks a lot.